All right. So this marks the beginning of my exam 3 written portion video. I wasn't able to find a whiteboard, find a study room with a whiteboard, and so my drawings will hopefully be sufficient. So let's get started with question number 1. So in this question, we're talking about human diseases that result from different chromosome deficiencies. And we're given a list and we're asked to say which of the terms is associated with a certain syndrome. So in A, it's which of the above terms is associated with the Patau syndrome. I don't know how to say that. And the Patau syndrome we know is trisomy of, I believe, trisomy 13. And so trisomy is one of the words that we put under there, and it's also aneuploidy. Because it's not all of the chromosomes that are replicated, it's just one, the 13 chromosome that is replicated. So it's trisomy, aneuploidy, and then it's duplication because that chromosome is duplicated. It's not duplication. It's just trisomy and aneuploidy. Then B, which of the above terms is associated with the conventional form of Down syndrome. So this isn't the familial form of Down syndrome, which happens by translocation, but that's not how this happens. This happens, it's a trisomy 21, and it's also a form of aneuploidy, and it happens from a non-disjunction in meiosis. And so those are the 3 terms. And then in C, which of the above terms is associated with Turner syndrome. So Turner syndrome is just one X chromosome, X and then 0, nothing else. And so that is considered monosomy, meaning that it's monosomic for that, meaning it's just one X chromosome. It has all the rest of the chromosomes, but it just, in the X and Y chromosome, it just has one X chromosome. And then it's also aneuploidy, and it's not euploidy where all of them are replicated. And then D, asks the Cri-du-Chat or whatever syndrome. And this happens from a deletion, I believe, on the 5 chromosome. And it happens from a deletion that results from an inversion and a translocation. So that's question 1. Sorry, those are just kind of ones you read off. Question 2, this one's a little more exciting. Hopefully you can read this. This talks about an E. coli DNA sequence, and we're going to be talking about like primers. There's 2 different primers, and we're supposed to indicate the polarity, and this is kind of just to understand conceptually how replication kind of takes place. And so I wrote out, these 2 lines right here are the DNA sequence that we're given, and this is our template strand. And so I couldn't fit it all in one line, so it kind of goes across 2. And then here's our 2 primers. And so what I first did was I wrote out the 2 primers and then wrote out the DNA sequence that would need to match up with these primers. And then I found them in the main sequence. And so this right here is for the primer A, and this is for primer B. And so we're supposed to indicate the polarity of the different primers. So for primer A, we know that since DNA is anti-parallel, that primer A would need to be 5' right here and 3' right here. So it's 5', 3'. Sorry, I'm trying to read that backwards. And then for primer B, I found that it was right here, and it's the same case. It's anti-parallel, and so it needs to be 5' and 3' right there. Okay, and then B, it asks, what is the DNA sequence that will be synthesized between the primers? And be sure to indicate the polarity. So this is the DNA sequence, and pretty much it just goes, since it synthesizes 5 to 3, because this is the 3' end right here, we synthesize this way to connect with this other primer, if that makes sense. And so these are the base pairs of that DNA sequence that is formed, and it's 5 to 3 because DNA is sequenced always 5 to 3. And yeah, and so it's in between these 2. I hope you can see that. Then C, it's which direction, right to left or left to right, is the replication fork moving as this daughter strand is being synthesized. And so this is an Okazaki fragment. And so we know that this is the lagging strand. It's on the bottom. And because it's on the bottom, we know that the helicase is unzipping the DNA from left to right. Or I'm sorry, from right to left. And so, wait, did I get that right? From right to left. So it's synthesizing this way. I'm sorry, I meant to say from left to right, since this is the bottom strand. So it's synthesizing from left to right because it's unzipping this way, if that makes sense. And so as it's unzipping this way, the leading strand up above is just kind of following the helicase, but the bottom strand, the lagging strand, is going, it has to go 5 to 3, but it can't go, yeah, it has to go in the 5 to 3 direction. And so that's why you need these primers on the bottom. And then D, it's asking which of these two primers was synthesized first, and it's primer A, because if you look at it on the bottom, on this bottom strand right here, the 3' to 5' end right here, if the helicase is unzipping it along this way, because that's what's kind of going on. He's going across this way. That means that primer A, this DNA sequence right here, was exposed first because the helicase made that available to put a primer on first before it got to B. So primer A is the one of the two primers that was synthesized first. Okay, and then 3. I'm going to have a hard time explaining this, but I'm going to give it my best shot. So this has to do with individual who's heterozygous, and they experience an inversion in one of their chromosomes. And so I drew a picture of how this inversion takes place. This is the normal one on top with the crossover between U and T right there. And then this is the inverted chromosome on the bottom. And so this one kind of reads like if you follow the line from here, and then instead of going straight across, it jumps. And this is the inversion spot, and then it ends normal right there. So all we're asked to do is find the 4 different results, the 4 total sister chromatids that result from meiosis if there's a crossover between U and T. So because this is an inverted chromosome, we know that there's going to be 4 different products, and we also know that it's inverted paracentric, P-A-R-A, because it's outside of the centromere. Because it's outside of the centromere, we know that when it goes through meiosis, there's going to be a dicentric chromosome, or dicentric sister chromatid, sorry, and then I think it's called an acentric sister chromatid. And what that means is one of them is going to have 2 centromeres, one of them is going to have no centromeres, 0, and then 2 of them are going to be normal, so are going to be just parental. So the first 2 gametes, one of them is the same parental one, just with that crossover between U and T, and the bottom one is the same case, just the same chromosome with that inversion that takes place right there. And then the bottom one, the way that you figure this out is you kind of follow this chromosome. So you go LMN centromere, OPQRS, and then, or I'm sorry, at this S where the inversion took place on the other chromosome, you have to switch over. And so, because that's what happens during this meiosis process. So you go S, T, U, V, W, and then because it's inverted, the chromosome follows along this way. And so then you have the rest of these, which is just R, I'm sorry, I can't read it. It's just RQPO centromere NML. And so this is the dicentric sister chromatid right there. You can see those 2 centromeres. And then this one, you kind of do the same process, but on the other side and start on the bottom strand. And so you'll go X, and then you switch over to the other chromosome at the inversion place, and that's S. And so you switch over really quick. You go X, and then S, and then UT because of the crossover, and then VWX. And it doesn't, there's no centromere in that one, so it's acentric. It's lacking a centromere. Okay, I hope that made sense. And so those are the 4 products of meiosis after that inversion. Okay, question number 4. This one's kind of fun too. It says, suppose that the DNA molecule shown below is a portion of an E. coli gene, and the E. coli gene is transcribed from right to left. Okay, so it's transcribed from right to left. Yeah, I'm sorry, it's hard to show this in the computer because it's like backwards. It's a very difficult concept to explain in a mirror. But anyways, so for A, you're asked which strand, the upper or lower strand, is the template DNA strand, and which is the coding. So I'm going to try to explain this. I understand this principle. So we know that the RNA polymerase, it follows along the template strand 3 to 5. It follows along 3 to 5, but it synthesizes 5 to 3, just like in DNA polymerase, RNA polymerase synthesizes 5 to 3. But it follows the template strand 3 to 5. And so looking at the template strands, whichever template strand from right to left, or I'm sorry, from right to left goes, yeah, from 3' to 5', that's the one that's going to be the template strand. And so it's this top one. From on the right side, it's 3', and on the left side, it's 5'. And so it follows this way. So this right here is the template strand, meaning that the other strand would be the coding strand, the bottom one. So upper is template, lower is coding. And then B, we're asked, what is the sequence of the mRNA molecule produced from this DNA molecule? So you start at the +1 site, because that means that transcription starts right there of the RNA molecule. So starting at the T, I didn't rewrite it, but you start at the T on the top one, because that's the +1 site, the thymine, nitrogenous base. You start there on that base pair, or at that base with T. And then you just go across from right to left. And that's important that you make sure you go right to left. And since it's RNA, wherever there's an adenine, you're going to match that with the uracil instead of a thymine. And so you get the following sequence, and you make sure it's 5 to 3 because that's what it's synthesizing. So you get this following sequence from the upper template strand. Then C, it asks which side the -10 and -35 sequences are located. So the -10 and -35 sequences are sequences that help in the process of initiation. Initiation or actual transcription hasn't started yet right there, but that's when the transcription factors start to come onto the DNA molecule so that they can begin transcription. So if that's the case, that means that it would need to happen before RNA started, or RNA polymerase started creating the mRNA or the pre-mRNA. I'm sorry, this is E. coli, so it's just RNA. And so that means it needed to start on this side. And so that means that the -10 and -35 sequences would need to be on this side of the transcription factor, or of the +1 site. And so the answer to that, it's on the right of the +1 site, on the right. So over here, if you see that little box. And then D, it's which direction on the coding strand. Which direction on the coding strand relative to the +1 site would the terminator be located? And this is on the left side, because the terminator is where it ends. And then that's essentially what all this question is asking. And the reason, so it asks about the coding strand and not the template strand for both C and D. The coding strand is antiparallel to the template strand, but it's still in the same, it's still matched up with each other. And so the coding strand would still have the terminator over here. because it matches up with where the template is being read. So yeah, that's the answer to the testing questions. I hope that made sense somewhat. Sorry I didn't have a whiteboard. But anyway, there's my video. Hope you enjoy it and enjoy your day. Bye.